Good morning, good afternoon, and good evening to all our attendees that we have from across the world. Welcome to Thai Global Health. My name is Dipty Desai, and I, along with Vani Parmar and Jessie Danjal, co-chair the Thai Global Health Initiative. This initiative was envisioned and brought to life by Venk Shukla, a Thai Global trustee and past president of Thai Silicon Valley. For those that are not familiar with Thai, it was formed about 28 years ago to foster entrepreneurship amongst the Indian population. Professional graduates seeking jobs in the US were well versed with technology, but not with business, and were hitting a glass ceiling. Thai provided a platform for them to start their own companies, become CEOs, get mentored, educated, funded, and allowed them to network with like-minded individuals. Today, CEOs and CIOs of many large enterprises are of Indian origin, thanks to the efforts of Thai. And Thai is now a global brand with 60 chapters across the world and a large presence in US and India. The mission and vision of Thai Global Health is to bring together Thai charter members from around the world that have an interest in health tech. The Global Health Initiative provides a platform that facilitates dialogue and interaction, exchange of knowledge and ideas, showcase of technology, and brings together investors, innovators, and enterprises. We would like to use this forum to study the innovative solutions from startups, understand the gaps from enterprises, learn where the money is flowing through investors, provide knowledge through mentors and advisors, and use the collective connections to create a heightened momentum, which can be tapped for the greater good for investing, mentoring, advising, and showcasing of ideas. Our talk today is focused on one such collaboration of partnership in AI and computational pathology for precision medicine in the context of oral cancer and breast cancer. The work we will hear from of today is through the collaborative efforts between 3 individuals and 3 institutions of high repute. Dr. Anant Madabhushi is the Donnell Institute Professor of Biomedical Engineering at Case Western Reserve University in Cleveland, Ohio. He's also the director of the university's Center for Computational Imaging and Personalized Diagnostics. You know, when I was talking to Dr. Anant, I realized that the 2 of us go way back and we attended the same primary school in Bombay, India. So, Dr. Shlomo Koyfman is the director of radiation oncology at the Cleveland Clinic Cancer Institute, and he specializes in head and neck cancer and radiation oncology programs. We will be hearing from him on his work on oral cancer and the use of AI technology for the therapeutic management of cancer. And then from all the way across the world, we have Dr. Vani Parmar. Dr. Vani Parmar is a professor in surgical oncology at the Tata Memorial Center in Mumbai, India. Dr. Parmar is an oncoplastic breast surgeon and has keen interest in 3D printing of organs. Currently, she's coordinating a number of trials in breast cancer across various centers in India. In the past, we've hosted talks on the use of AI for diagnostics. Today's talk is a little different. It's focused on the development of novel AI tools in conjunction with biopsy images for oral cancer and breast cancer. So it's more on a therapeutic management side, right? That will allow for risk stratification of the disease. So this population segmentation will help us identify patients that can avoid aggressive therapies like chemotherapy. So this should be of great interest, and it's a little different flavor than most of the talks that you've heard in the past. We do want this to be an interactive. Those of you that are listening to the talk, please, we would love to have your questions. Please post the questions in the Q&A box that is down below, and we will have a Q&A session at the end of the talks. So for the next 30 minutes or more, let's hear from Dr. Madabhushi, Dr. Koyfman, Dr. Parmar on the use of AI in the therapeutic management of cancer. Thank you, and over to you, Dr. Madabhushi.
Thank you so much, Dipty. Real pleasure and honor to be joining everyone this morning for this Thai Silicon Valley event, and to talk about the work that our group has been doing in conjunction with Dr. Parmar and Dr. Koyfman. So I just want to maybe introduce myself and my group a little bit and lay the context a little bit for this particular panel discussion, which you, I think, correctly identified, Dipty, is, we hear a lot about artificial intelligence today, but a lot of it is really in the diagnostic setting. A lot of it is on how can AI be used with CT scans or MRI scans to help the radiologist do better diagnosis. How can AI be used with tissue, with pathology images to help the pathologist make a better diagnosis, to identify the presence of cancer from a pathology slide. The main focus of our group at Case Western Reserve University in Cleveland, Ohio, has been a little different. And so while we're developing a lot of novel artificial intelligence technologies for doing disease diagnosis, identifying the presence or absence of cancers for lung, for breast, for brain tumors, from medical imaging scans, a big part of our focus is really on developing tools that will aid the clinician, that will aid the oncologist on better treatment decision-making, on prognosticating outcome for patients with cancers. Because the fact is that today, and these are US-centric statistics, but my sense is that these statistics are fairly consistent and similar across the world. 40% of the adult population in the United States will be diagnosed with some form of cancer in their lifetime. And that's a massive, staggering statistic, which means that, yes, early diagnosis is important, but given the number of cancers that are going to be identified, there really is a major issue and a major challenge on how to manage, how to treat these patients. Now, we know in many different diseases that overtreatment and overdiagnosis is a major issue. We know that a number of patients who have potentially less aggressive cancer tend to get treated very, very aggressively. Now, today we're not going to talk about prostate cancer, we're going to talk about oral cancer and breast cancer, but prostate cancer is a classic case in point of a cancer that's treated extremely aggressively. 1 in 40 men in the US will be diagnosed with prostate cancer in his lifetime, and only 1 in 6 will actually die on account of it. And yet we treat these cancers very aggressively. We sort of throw the proverbial kitchen sink at a lot of these cancers. And so our group's focus has really been on how can we develop novel AI technologies that work with routinely acquired data, right? So any cancer patient, any cancer diagnosis done anywhere still relies on pathology, relies on biopsy, relies on tissue images. And so our group has paid particular attention and focus on using AI technologies, developing and applying AI technologies with the biopsy images, with the tissue images, to go in and find features, to find patterns of cells, cellular arrangement that can tell us more than can be visually discerned by a pathologist. So in other words, going beyond just disease diagnosis, going beyond just grading of the disease or staging of the disease, to try to find patterns, to try to find features that can actually tell us about what the outcome for a patient's cancer might be. Is this a cancer that is going to have a recurrence in 5 years? Is this a cancer that's going to metastasize? And therefore, is this a cancer that you need to treat more aggressively, that is with chemotherapy, for instance? Or is this potentially a cancer where one could help de-escalate the therapy, one could de-intensify the therapy, one could potentially obviate the need for chemotherapy in some of these patients? Because like I said, the fact is that we are over-diagnosing and over-treating a large number of cancers. And I'm a lowly bioengineer, so don't take my word for it. You'll hear from Dr. Parmar and Dr. Koyfman, who are clinicians, who can attest to this issue. Now, one of the other things that my group has been particularly interested in is having impact, right? Having global impact in the context of cancer. What is it that we can do with technology to develop technology that really can have an impact to the maximum number of people in a maximum number of countries around the world? Now, I'm a Bombay-wala myself. I grew up in Bombay. And so one of the things that's sort of very near and dear to me is how do you develop technologies that are non-disruptive and really could have an impact in low and middle income countries like India, where you don't quite have the same kind of resources, You don't have the sort of reimbursement structures that you might have in the United States. And so our group has very deliberately tried to look at what is the ubiquitous technology, what is the sort of lowest hanging, what is the common denominator, right, that really unifies, connects cancer patients across the world. And like I said, it's the tissue slide, it's the biopsy slide, it's the pathology slide that a pathologist uses to identify the presence of cancer. However, now with the ability to digitize glass slides, we can actually convert these glass tissue slides into high resolution digital images. Now, the benefit of that is with the digitization of glass slides, computational technologies, AI technologies can be brought to bear on these glass slides. And we can start to mine these really subtle patterns relating to the arrangement of cancer cells, arrangement of immune cells on these glass slides, that we can then start to connect to disease outcome, that we can connect to therapeutic response. But the beauty is that we are not destroying any tissue, right? There are other technologies out there that essentially involve destructive testing of the tissue that are based on what are called molecular diagnostics to try to identify whether somebody would need chemotherapy or could avoid chemotherapy. But these tests are expensive. Now, they're available in the United States, they're available in Europe. They cost several thousands of dollars. And the beauty with the kind of technologies that we're looking at is that it doesn't break the bank. You really just require an image of a tissue slide. And then it's the application of these AI algorithms, these computational algorithms, to these glass slides to be able to render a prediction of outcome or a prediction of who is going to benefit from therapy. And so this is another important key component here, is being able to develop technologies and apply technologies that are amenable and affordable in low middle income and low resource settings where you don't have access to the kind of resources you might in the US, and therefore may not be able to afford the more expensive molecular diagnostic kind of tests. So with that very brief introduction, what I would like to do is pass the baton on to my dear friend and colleague and collaborator, Dr. Vani Parmar, who's a breast surgeon at the Tata Cancer Center in Mumbai. Dr. Parmar and I have been working for a couple of years now on application of these technologies for breast cancer. So without further ado, then I'll hand it over to Vani to talk about the implications of these technologies for breast cancer prognosis.
Right. Thank you very much, Anant. And that was, I think, a beautifully set stage for us to take the discussion forward. As Anant has very expressively described onto the application of AI and how it takes us forward. Let me give you a brief on what is it that we see in India and why did we think that this particular technology had an important application in our patients. So there is no screening in India. So that's the first background on which I'm going to start, because what we see is a little different kind of cancers as compared to the cancers, breast cancers that we see in the West. And knowing fully well that the cancers that we are seeing are slightly larger, we did look at the biology of these tumors and cancers. And I'll give you a brief, so maybe it'll give you an idea onto what exactly are we talking about. So the number of new cancer cases that we see in India is about 160,000 annually, new cancer cases. Half of these, that would be around 80,000 of them, would be early breast cancers. So you're talking about cancers that, these are cancers not screen detected, but more of, as they present to us clinically. So I'm a breast cancer surgeon, and I'm going to talk about mostly the surgical aspects of it. And of course, it's not just the surgical aspect, because we also do the planning of treatment for these patients comprehensively in a multidisciplinary team, which comprises of radiation oncologists, medical oncologists, as well as pathologists. So when we see these early cancer cases, half of these being node positive, they would definitely require most of the multimodality treatment that we just spoke about, in the sense of we do the surgery, and followed by surgery, we will have an adjuvant chemotherapy as well as radiation therapy that we cannot be avoiding in these patients. But then there is another set of patients where the risk may be actually low. Currently, because the available tools that are used for identifying the risk of these patients for recurrences, these tools are expensive tools based upon molecular tests, and they require the tissue to be processed completely. And then when we do get the report, it's usually a high expense that we are incurring in finding out the outcomes, or rather the prediction of sensitivity to treatment. So we are actually looking at predictions to decide prognosis. Now, these are two terminologies which we will be using very commonly when we talk about cancers. So the tools help us to predict whether or not the cancer responds to a treatment. Now, that is not really a right kind of a tool, because we're just looking at prognostic outcomes and deciding the prediction. But interestingly, it has shown us that one can identify correctly certain tumors which we may be ending up overtreating. And when I say tumors overtreating, it's actually the patient getting the treatment. And the toxicity of most of these anti-cancer drugs and anti-cancer treatments are so high that we will probably end up having less of benefit and more of toxicity. And this is where I think AI has really helped us to identify such patients. What we are working together with Anant is actually getting us to identify patients who are biologically low risk, and these are the ones who express ER and PR receptors and not expressing a HER2 receptor, which is indicative of aggressive biology. And keeping this in mind, we actually have as much as 30,000 cases maybe annually in India. And these are the patients who probably will end up being correctly identified as those who would benefit versus those who would not benefit from chemotherapy by using such a technology. Not only that, we would be able to utilize uh, images, digital images, scanned images of slides, and hence no tissue would be actually utilized for getting the results of the tests, and we would be able to use minimal amount of tissue for such a test. In India, cancers are definitely of a higher grade. What we see, younger women, so the median age here is about 50 as compared to a decade older in the West. The risks of cancer is in the developing countries like India is actually lower. It's 1/4 of what we see in the West. But since the population is so large, we have much larger population of breast cancer patients. And that's the reason why a small contribution of such a kind would actually save many more women from receiving unnecessary and over treatment in the form of chemotherapy. So I think I will let Shlomo do the rest of the talking right now, because we will have more discussions ahead. So.
Thank you, Vani. Yeah, Shlomo?
Thank you, Vani. Thank you, Anant. And thank you to TIES for the invitation. It's a really cool thing to have you guys kind of give us voice, because our frustration is what we manage in clinic. But of course, we don't have the tools to really design the molecular diagnostics and the AI diagnostics and predictive tools that are going to change our practice. That's why we need industry collaboration. So you've heard a lot about the idea that we overtreat a lot of people and that we need to reduce the treatment in a lot. And that's true. I want to talk about a totally different problem, okay? I want to talk about the problem of how often we walk into a room, we give them the best we can give them. We give them chemo, radiation, surgery, whatever we give them, and it still doesn't work, and the cancer comes back and people die, okay? And that's a huge problem. And I would argue it's probably a bigger problem than de-intensification. And the issue is this, you know, Anant and Vani talked about breast cancer for a bit, and breast cancer is one of the rare cancers that actually has a lot of biomarker driven care. There's something called oncotype. There are these tissue related tools that can help stratify patients. I think one of the most important things I want you guys to take away is most cancers don't have that. We're still treating cancer like it's 1985 most of the time. My practice hasn't changed in 30 years of medicine. We've gotten fancier, we've gotten a couple new drugs, but it's nothing great. Progress in cancer in some ways has been revolutionary in terms of developing new agents, the advent of biological therapies, the advent of immunotherapies. These are major explosions. But those are usually used in the end in the non-curative setting. In terms of curing people, other than screening, which has been great, we're not making huge dents. And the reason is because we have a fundamental problem in cancer. And the fundamental problem is that when we do research and when we do trials and when we try to figure out, hey, how do we do better with these people? We don't have sophisticated ways to bucket patients properly. So I'll give you a perfect example. Let's say oral cavity cancer. It's one of the most common cancers in India. It's a terribly deadly cancer in India. In the US also, of all the head and neck cancers I treat, it's probably one of the worst. And probably 50 to 60% of people walk in with locally advanced disease, and we cure maybe about half of them. But we got a lot of people, and younger people are getting this. People who never smoked are getting this more often. And they'll get a tongue cancer and it'll look like a no big deal stage 2 cancer. We'll cut it out. We'll do good surgery. Sometimes we'll even add radiation, which is what I do because there are some pathologic features we don't like, and then it'll still come back. And we have things at our disposal. We have chemotherapies, we have immunotherapies. But the problem is if only 20 or 30% of the time it's coming back, we don't have the tools to know who those people are. If we could identify who are those patients most likely to fail, then we can actually study them properly. So we just did a major national 700 patient trial in head and neck cancer, comparing radiation by itself to radiation with an antibody drug called cetuximab. Irrelevant. The point is, it's probably going to be a negative study and it's not going to help. Now, is it because it's a useless drug? No, it's FDA approved in head and neck cancer. We know it works. But the problem is within those 700 patients, maybe 50 would benefit, maybe 70 would benefit. But since we have no idea who those people are, we just give it to tons of people. And, unless you have a huge signal to noise, efficacy signal, you lose it, and you lose that benefit awash the statistical noise. What Anant's technology is so beautiful about is, in addition to the fact that it's just a digital path slide that you can take a digital image of anywhere in the middle of, some cruise ship and still get all the data you need, is that, when I met Anant, he already had an R1 in breast cancer. He was already looking at lung cancer, and he came to me in oropharynx cancer. And then I said, what about oral cancer? He's like, sure, let's do that. It's a platform that can be applied to all cancers. There are no biomarkers in head and neck cancer. We don't have an oncotype. We don't have anything. The only thing we have is whether you have virally, that's it, which is a small amount of our cancers. So the power of this tool, and what I'm doing, is how do I find the people that have intermediate risk that right now we're just giving surgery and radiation to, and we've done trials that show there's no benefit to chemotherapy, there's no benefit to immune therapy, but how do I pick out those people that are most likely to fail and study them? And if I can do that, that's the idea. If you can take 100 patients and say, well, actually, these 100 patients really are a mixture of 3 categories. There's a really low risk patient here. They probably didn't even need the radiation. And there's an intermediate that the surgery and radiation is just fine. And then there's another third that this isn't enough for. We're blind to that now. We treat them all the same. They're all 100 getting surgery and radiation. This is kind of the power of this predictive assay. Because if Anant can discover the signatures, again, what the cells look like under the microscope, how large they are, how they look, which a pathologist's eye could never see, that are implications to biology, then what we're able to do is bucket those patients properly, study them more rationally, discover the therapeutics that are going to do better for them, and ultimately probably lead to increased cure rates. And that's why I'm a clinical trialist. I'm running a Merck study in skin cancer with immunotherapy, and we have other studies in RTOG looking at all kinds of different chemotherapy combinations, new biologic therapies. But most of these studies are going to be negative because most of them are in unselected patient populations. And I think that's the biggest power of this in any cancer. You give Anant enough slides in any cancer and he'll do the bioinformatics and he'll find a signal, because we know every single cancer follows the same paradigm. We stage people in groups of stage 3 or stage 1, but we know they're biologically heterogeneous, and we don't have the kind of assays in probably 90% of cancers to figure that out. And this platform is powerful because it can help sort that out. Now, again, I will say, academics, I love this, but this isn't the kind of easy work that R01 funded scientists sitting in a lab are going to be able to just bring to clinic. You need industry partnerships to discover it, test it in a rapid way, and really scale it out. So that's the opportunity, and I think it's a really exciting one for people like me and Vani, who have to walk into these patients' room and walk out saying, we did whatever we could and we failed. And how do we get better at it? So that's why I think it's a potential paradigm shift in the industry.
If I may just add, Anand. So exactly what Shlomo has just been talking about. For years and for months, we have just been treating breast cancer based upon certain clinical features and the basic histopathology slides and the reports that we get, routine histopathology slides. We have really not been very wrong in predicting as to what would be the right kind of treatment for them. But yes, there is a segment of patients that we end up over-treating. What Anant has actually shown us is that we can use these in a much more detailed manner by taking out the subjectivity that happens because of individuals looking at these slides and coming back with, I feel this looks to be a little more hormone sensitive or hormone insensitive. So what we are looking is objectivity of what you're seeing in a routine histopathology slide, bringing it down to a level that it becomes much more disciplined, and there is the room for room for errors is actually reduced, and you are able to get precision into the way you look at a tumor and its characteristic. And it's actually brought back the importance of the phenotype and not just the genotype. So that's where it's very important, because the phenotype actually speaks a lot. And what we are trying to do in this is probably bringing up the phenotype of a tumor and trying to help a tool, make a tool that will help us to decide as to how this tumor is actually going to behave. And I guess it's really working out well, because the results as we see them in the last analysis that was carried out in a set of our patients has clearly shown the divide between the high risk and the low risk. And that's done at absolutely, I would say, no cost right now, because there's not much that really goes into it, excepting, of course, the technology itself that's being built up. And if you look at the Oncotype DX, which is the available tool, it's completely out of reach of majority of Indian population. So I think this is an extremely promising tool for us to use in the clinic.
So if I could just add, I think that was a really beautiful summary of the two kinds of problems. And thank you, Shlomo, for providing sort of the other side of the spectrum, where on one end there's the overtreatment and trying to prevent unnecessary chemotherapy in a lot of cancer patients. But at the same time, and I think this is one of the questions that just popped up in the Q&A box, is what about those patients who don't respond to therapy? What are those patients who potentially are not getting the kind of treatment that they require, that Dr. Koyfman just talked about? And that's the opportunity. So with better quantitative computerized analytics applied to routinely acquired data, pathology images, we have the opportunity to provide more granular risk stratification for a cancer patient, right? So in other words, we're able to now come up with a risk score that says, okay, for this cancer patient, this is the aggressiveness level, right? And so you can look at that spectrum of aggressiveness, and based on that spectrum of aggressiveness, you now have a sense of how much more aggressively you need to treat the patient. It may be that the risk score, the aggressiveness score, based off the features that we're mining using AI from the pathology images, are very low, and therefore that's a patient who's a candidate for de-intensification or avoiding chemotherapy. But if a given patient has a high risk score, then that's clear indication this is a person who probably needs more aggressive therapy. In fact, some work that we're doing currently with Dr. Koyfman shows that the current staging criteria, that's essentially the way to compartmentalize and bucket patients into different risk categories, is woefully inadequate. So what we did recently was we looked at a series of oral cavity cancer patients with Dr. Koyfman and found that in patients who were stratified under AJCC stage 1, so stage 1, this is all based of pathologic assessment of the tissue, those patients under current protocols, or under current guidelines, will get surgery alone. But our analysis is saying that in that set of patients, there are some patients who clearly are going to do worse. They have a much higher risk score. And that's exactly to Dr. Koyfman's point. That's the data that he would use to say that, okay, the current status quo is give all of them surgery, but this test is saying that some subset of them should be getting more aggressive therapy, should be getting radiation therapy over and above surgery. And so that's the value add. So one hand, in the context of breast cancer with, working with Dr. Parmar, there's the opportunity to obviate the need for chemotherapy in some subset of patients. At the same time, in oral cavity squamous cell cancers, there's an opportunity to identify the patients who, we go beyond the status quo and potentially give more aggressive therapy to. The one other quick thing I want to just talk about is technology. I know some of it's been said, it's digital, a base of computational analysis of imaging data. And that's actually provided a unique opportunity working with Dr. Koyfman as well as working with Dr. Parmar that allows us to take advantage of retrospective data, right? This piece cannot be overstated. We've talked about, the fact that it's digital, it doesn't destroy tissue, it can be low cost. But the real key advantage of the fact of technology like this is that it allows us to go back 20 years, 30 years to completed clinical trials from the 90s, access the slides from back in the 90s where these clinical trials were done, digitize those slides and analyze those slides. So in some work that we're currently doing with a clinical trial that was done in the mid 90s for breast cancer. And we've actually demonstrated that for those women that were treated, 30 years ago, 25, 30 years ago, that this risk predictor that we've developed today can be validated on women who've been treated 25 years ago. So that's the beauty of the technology. You don't necessarily need to validate it solely in a prospective way, which I think is a lot of the hestation for new biomarkers, is that you need to validate it prospectively. The beauty here is you can go back retrospectively and validate on patients, who'd had a treatment done, 20 years ago, 25 years ago. And there, the beauty is also, exactly what happened to the patient. You've got the long-term outcome. And so it provides an opportunity to validate the tool in a very robust way.
Absolutely.
So thank you, Dr. Vani and Dr. Anant. And I think, do we still have Dr. Shlomo? Maybe, I know he was having some trouble with his connection, but
he's also in the clinic right now, so he's wearing proverbially two hats right now, so he might be sort of in and out.
Yeah. All right. So no, truly interesting, and I know that this would be of great interest because just a couple of days ago, I had posted, a thought in our group, which is about 200 to 300 people, the whole Thai Global Health Initiative group, which is comprised of charter members from Thai that are interested in healthcare. And we had said, hey, how many of you guys would be interested in looking at new startups that are doing work in using technology in healthcare? And also, how many of you are investors that are looking to evaluate these technologies? And there were a lot of hands that went up. So there's great interest in that group to learn about new technologies, what they're doing, and potentially invest in them. This is probably one of those kinds of technologies. And like Dr. really needed to be just addressing oral cancer and breast cancer. Those are just areas that you decided to focus on for now, but it could be literally expanded to any of the areas. And whether you have a biomarker or not, like in breast cancer you do, with oral cancer, Dr. Shlomo mentioned that you don't have. So it's great. A couple of questions that are coming up, and one of them I think you probably already addressed, but I'll just bring it out again. So from the conversations here, we see that AI, although a new technology, plays a very important role to help in clinical decision-making for treatment of oral and breast cancer. And we've talked in the past about radiology diagnosis and pathology diagnosis using AI. So could you again bring out the nuance on how is this being this therapeutic management, how is it different from the AI for radiology diagnosis and pathology diagnosis? Maybe you can just illustrate those.
Sure. Yeah. So, one of the things here with, you know, so if you think of the continuum, right? So now, Dr. Parmar is a breast surgeon. Dr. Koyfman is a radiation oncologist. So really, when they come into play, in a manner of speaking, when they come into play is really after the diagnosis of the presence of disease has been made, right? And so right now, the use of AI, there's a lot of focus on that initial step, right? So essentially looking at medical imaging scans, looking at CAT scans, looking at MRI scans, or looking at pathology images to help with the detection of the disease, to be able to tell, is there cancer present on a CT scan? Is there cancer present on a pathology slide? To help with rendering that initial cancer diagnosis. What we're doing with Dr. Parmar and Dr. Koyfman is taking that same data. We're taking the pathology images, but we're saying, okay, we have a definitive diagnosis of cancer. Now let's look at these cancer slides. Let's try to find features or patterns on these slides and say, what is the aggressiveness potential? What is the likelihood that this disease is going to progress? What is the likelihood that this disease is going to metastasize and get worse? And therefore provide cues now for Dr. Parmar and Dr. Koyfman. It's a companion diagnostic. It's a decision support tool for the clinician on how to better, manage the treatment strategies for the patient. And like Dr. Koyfman said, this is a platform technology. We're talking about oral cancer, we're talking about breast cancer here. I see a question here on, about prostate cancer. Saying, 1 in 6 men will die if we don't treat more aggressively. That's absolutely right. And this is to Dr. Koyfman's point, that it's not always about the de-intensification. We know that certain cancers are more aggressive, but the challenge is still being able to identify those more aggressive cancers that could then benefit from more aggressive therapy. And prostate cancer that I mentioned, 1 in 6 men will get it, 1 in 40 will die on account of it. Well, we don't want 1 in 40 men dying because of prostate cancer. The question is, how do we identify those men? Because those are the men who could benefit from adjuvant therapy after surgery. So a lot of them get radical prostatectomy, they'll get surgery for removal of the prostate. But we know that some of these men are going to have a recurrence of the cancer. The cancer is going to come back, it's going to metastasize. What we're finding now is by doing our analysis on the surgically excised specimens that we can identify which of these prostate cancers are going to come back and therefore could benefit from chemotherapy or radiation therapy after surgery. So we can intensify the treatment. We can identify those candidates, those patients who could truly benefit from intensification of treatment. The other question is about toxicity. And that's a very, very valid question about response to toxicity. And a lot of the work that we're doing right now is trying to address this issue. Brain tumors is one particular area, and I'd love for Dr. Koyfman to chime in as well. Brain tumors is an area where we treat with chemotherapy, we treat with radiation. Once the patient has been treated, they come back, they get an MRI scan. Very often, it's very difficult to tell what's going on. There's something that shows up on the MRI scan. Is it because the cancer's come back? Or is it because of the radiation treatment? We know that a lot of cancer patients will have something called radiation necrosis, right? And that looks very, very similar to the cancer on imaging. Some of the work that's happening in our group is showing that we can actually distinguish between radiation necrosis and the recurrence of the cancer. And that's important because that way we can prevent unnecessary interventions in these patients to figure out what it is. So the opportunity is also in being able to identify the toxicity for, in a lot of these patients, because they will have side effects of the treatment that will manifest in these patients. And there are other issues as well. I mean, immunotherapy that Dr. Koyfman mentioned, has changed the treatment landscape for a lot of cancers. But even there, the challenge is that, only 25% of the time does immunotherapy work. 75% of the time for lung cancers, for instance, it may not work. And these are expensive treatments. We talk about chemotherapy being expensive. Immunotherapy is $250,000 per patient per year. If 75% of patients are not going to respond, well, that's a massive financial toxicity associated with treatment that's not going to work. So we're looking at trying to address those questions. How can artificial intelligence play a role in identifying those patients who are not going to benefit or respond to this very expensive therapy. And it doesn't mean that they don't have other options, right? I mean, we still have chemotherapy. So if immunotherapy is not going to work, and if you could identify that in advance, then maybe we can identify which patients could avoid immunotherapy, but instead maybe get chemotherapy or get some other combination of chemo and immunotherapy.
And I wanted to add one thing. I see there's a question on the role of multi-omics biomarkers in patient stratification. So it's really sexy. It sounds awesome. It's fabulous. We don't do it, and we're not doing it anytime soon, because the problem is that you have 2 minutes to go about. You have two ways to go about doing this. You either try to figure out the genes that universally predict breast cancer outcomes, and you kind of focus on that, or you try to drill down on an individual's own unique cancer genotype and phenotype and have their own personal genetics kind of influence how you make treatment decisions. The latter kind of makes more sense, but it's just logistically unfeasible, cost prohibitive. The labor to get it to sequence a person's tumor. Is it the tumor? Is it the RNA expression? Is it the protein expression? I mean, this is what the scientists are working with, but that's nowhere near clinical utility yet in any kind of scalable way. And on the front end, it's obviously like, well, if you just bucket everybody together, and look at 20 genes, 30 genes, whatever, that's going to be a limited way of looking at things. I think the nice thing about this platform is it's actually agnostic to the genes per se. It's agnostic to the RNA expression or even the proteins. It's the final product phenotype of the cancer. I can't tell you how often I'm sitting in a tumor board, and we're sitting 25 of us in a room. We will today at 4:00, and our pathologists, we're looking under the microscope, and they say, this is a moderately differentiated squamous cell cancer of the oral tongue. This is a moderately differentiated squamous cell cancer of the oral tongue. And I'm sitting here looking at the thing, and I'm like, okay, I'm not a pathologist. I don't know what I'm talking about, but I'm telling you, these are not the two of the same things. But they said, yeah, but I mean, based on our categorization, like that's just, they fall into the same bucket. But they're not, because you can see it with your eyes. What you see on the pathology slide is the culmination of a lot of the genetics, et cetera, in the phenotype. Now, we're blind to some of the nuances on that, but Anant's kind of hypothesis is all kind of features that we don't even think about, how connected they are, their spatial relationships, how big the nucleus is to the cytoplasm. I mean, stuff we don't even think about have clues in them that are lending to the biology. And I think that's the beauty of this technology, is it's much more kind of individual patients can their own phenotype, and there's an infinite number of possible combinations that AI can account for that a gene expression profile just won't. You're going to say, is this gene upregulated, yes or no? And we know cancer is very heterogeneous and a little bit complex. The other thing I would say is, somebody asked the question about, is this just a one-time thing at the beginning of treatment, or could you use it in follow-up to help you kind of figure out subsequent therapies once they fail, what the next step is, et cetera? At this point, we're kind of validating it in the upfront setting. But there's a growing trend. Let's say we treat a cancer and a year and a half later, it comes back in the neck or it comes back in the lung. A lot of times the way we would, we might biopsy the tumor just to confirm that it was cancer. But if you had a genetic screen on it a year before, you'd use that and assume it's the same. We now know that things are changing, and there's a lot of work being done. Hey, when it recurs a year and a half later, or when a lesion pops up that's different, biopsy it. Its genetics may have changed. What it looks like under the microscope may have changed. Its behavior may have changed. And so in theory, this is a crazy cheap way, as Anant says, all cancer boils down to your biopsy. In theory, this is a way that if you're ever rebiopsying the person at recurrence, run it through the test again and see what you come up with. There's no reason why this technology wouldn't be able to say, hey, should we give them the carbotaxol regimen? Should we give them the pembrolizumab, or should we give them the combination of the two? There was just a major trial on head and neck cancer. The KEYNOTE-048 trial totally changed the paradigm. Nobody has any idea what to do now because we're not sure. Do we give all three? Do we just give chemo? Some get better with immunotherapy. We just don't have the biomarkers to help drive clinical decision-making in a rational way. So I think that right now, the stage at where this is in the discovery stage. We're trying to figure out what these signatures are and validate them on large randomized trials that we do. You know, for example, I'm part of the NRG cooperative in head and neck cancer. We run 600, 700, 800 patient trials in all the diseases we treat, and the tissue just sits in a biorepository in San Francisco, and nobody's done anything with it for 25 years. You know, we've connected Anant with them, and we're going to be looking at all those slides and validating it on these people that were already treated and we have their known outcomes. And that's kind of the pipeline to validate it. And then once it's validated and it gets into clinical use, then we might be able to expand its indications in some of the ways we've been talking about.
Truly fascinating. Vani, did you want to add something here? Actually,
So you know, the genotype that's available, the Oncotype DX test and the tests that are currently available, the molecular tests, they do tell us the presence or absence of a gene, but the actual expression of any of them would be the one which would impact on a patient. So when we're talking about all these terminologies, we are finally looking at the beneficiary at the end of it is a patient. Just having a gene or being able to pick up a gene does not mean that the gene is actually functional. And what this does is what the AI does and what it is looking at, what Anant is trying to improve for all of us is to find out the functionality of these. See, unless and until the phenotype expresses what the genotype says, it really is not going to be something that's going to impact on the outcomes. And the treatment that we're offering is going to work only if that gene is expressed. So just having an gene within the whole plethora does not mean that we're going to have an outcome that's going to be beneficial. And that's where it really helps us, because we may be coming up with newer drugs, newer medicines, newer treatments, more toxic, but we will not be able to know whether or not this particular patient is going to be benefiting or not. And what we're seeing is the actual expression. So I think that's where it makes a big difference.
And the beauty is that all you need is a slide, right? In this, you don't need expensive equipment and expensive tests, or in this case, genomic analysis, in order to come to the conclusions that you're coming to. And
Absolutely.
those slides already exist out there.
And that's the beauty of it, because you're really looking at something that's not going to be wasting away tissue, testing, going on repeatedly testing, especially in recurrences, etc. We may not even get enough of tissue to be able to process them through such tests. And what we're looking at is really using just an image, and a virtual image is being used to analyze and give us an answer, which probably the whole tissue is not able to give us. And that's the beauty of the whole test. And that's what really attracts and appears much more applicable to, especially in our set in the lower and middle income countries, where we do have a limitation of the amount of expenses that you can put in for them and the tissue that can be brought in. And if you look at it, it's a big country. I mean, to be able to get the tissue across to a central lab and get this processed itself is a cost that we're going to be just, completely removing from the whole armamentarium. Yeah.
And it's also not just cost, it's the logistics. I mean, a lot of these decisions have to be made in a week or two. Time to treat is kind of a big deal. From the time surgery happens, the final pathology report comes 3 weeks later. You want to get them started within 6 weeks, or you have a decrement in outcomes. A lot of times in the metastatic setting, they're sending Tempest and next generation sequencing. It can take 2, 3, 4 weeks to get results, which in that setting is fine. But we're talking about upfront curative management decisions. You don't have that kind of time. Whereas, you can take a picture of a slide, send it over, get a result in, a couple of days.
Yeah, so if I could just jump in, I mean, as a bioengineer, this is what you live for, right? You get your clinical collaborators even more excited about the technology than you are in developing the technology. So, you're doing something right. So I just want to add a few more things. We talked about, genomics and such, and gene expression and so on. There are a whole bunch of other fascinating things that we're discovering with this technology. I just want to do a quick shout out to some of the work that we're doing with Dr. Parmar, this collaboration with Tata Cancer. We started to work on this, area of breast cancer several years ago, but, with Dr. Parmar working at Tata. One of the things that we've now started to look at is, are there potentially subtle differences in the cell arrangement between breast cancer patients in India versus breast cancer patients in North America? And we just had an abstract that we jointly had accepted at the San Antonio breast cancer meeting that actually suggests that there are differences. We were actually able to use our AI approaches to find that the risk features, the features of the morphology, were subtly different between South Asian women with breast cancer versus North American women with breast cancer. Why is this important? What is the reason? What is this telling us? Well, it's telling us that potentially one may need to think about creating population-specific risk models. That is, you could create one sort of generic, Oncotype DX has been mentioned several times. Now that's sort of a one single test that was created about 20 years ago because it was dependent on tissue. Well, the tissue that it was developed with, that tissue doesn't exist anymore. So unfortunately, that test is stuck the way it is. It can never be changed. The beauty with what we're doing is as we identify new features, new patterns, as we make new discoveries, for instance, in this particular case, that there are subtle differences in the appearance of breast cancer between different populations, we can start to stand up population-specific risk models that are more closely aligned with a particular population. And this also stems from other work that we've done in other diseases. For instance, we've looked at prostate cancer and published some work earlier this year, where we found that if you look at African-American men in North America with prostate cancer versus Caucasian-American men with prostate cancer in the US, there were significant differences in the appearance of the disease phenotype between these two populations. And so what we were able to do was to create a specific risk model, particularly for African-American men, and found that that population-specific model was much more accurate in predicting outcome and predicting recurrence risk in African-American men compared to a model that was population agnostic that didn't really account for these differences. One of the other questions that also came up is about the status and where we are currently with the technology. Like Dr. Koyfman mentioned, we're still in the process of validating these technologies. And in fact, just interestingly, just this morning, literally 2 hours ago, we just got an email from NRG just giving us access to one of their clinical trial data sets, collaboration that's, I want to thank Shlomo for having engendered
nice. That's
all
good
of these
news.
partnerships. But this is the beauty of this technology, is that you can go back and validate it in a very robust way of retrospective data sets. The critical piece is you need to validate it on randomized clinical trial data sets. Again, the beauty is these have been done, these exist. We don't want any tissue, we just want the slides. The slides exist. Once they get digitized, we're off to the races. And so where we stand right now is we continue to validate this technology, we continue to generate the data. This just happened by chance. It wasn't intentional, but in fact, just later this afternoon, we have 2 abstracts going out from the group. One on using AI, validating for the first time that the technology actually works very nicely, very compellingly in breast cancer patients at the Tata Cancer Center. This is work we're doing with Dr. Parmar. That work is going out later this afternoon. And similarly, later this afternoon, we have a large study with over 1,000 patients with Dr. Koyfman in head and neck cancer, where again, we're demonstrating very nicely that in each of those different stage groups that I mentioned, stage 1, stage 2, stage 3, that this technology provides additional granular information. It is able to identify high-risk patients, even within these individual stage groups. And like I said before, right now the status quo is just treat them in sort of almost a monolithic way within each of those compartments. So everybody in a stage 1 gets treated the same way. What we're showing very nicely in over 1,000 patients now is that even within stage 1, there is a high-risk group and there's a low-risk group, and similarly in stage 2 and similarly in stage 3. So we're providing more granularity. And I think this is what we are looking to continue to do. But again, this is just a quick shout out to the TIES community and to those interested. And we're still very much aware that ultimately to take this, to move this to clinical practice, we need to work with the entrepreneurs among you to partner together. We're doing the research, we're working with amazing clinical collaborators here, we're getting access to the data sets. But it still needs the entrepreneurially minded to help move this to the next level, to translate this, to essentially get it into a product form so that then this can be used by Dr. Parmar, that this can be used by Dr. Koyfman, because clearly you can sense the excitement and the interest here. But as a lowly academic, we're doing the research, but it requires collaboration with the entrepreneurial community to take it to the next level, to really translate this into a clinically deployable test.
So I just want to add to that, moving any of these tests from the bench to bedside, as we would call it, it's very important to run it through certain clinical tests as well as clinical trials. And it does need to be applied to a large number of patients. And that really requires an intensive research and intensive monitoring. Of course, it's shown us very interestingly in the study that we carried out and the set of patients that we did apply the test to, that in spite of having a very wide variation in the clinical presentation, just simply if you look at it, we had such high-grade tumors as compared to the ones in the West. And we also had tumors which were much larger as compared to the ones that we see in the West. We had more of younger patients as compared to those in the West. But the test cleanly divided them into the low risk and high risk. And that sounds extremely promising. That means that irrespective of what kind of patient you're looking at, you're applying the test and you're getting an outcome which would translate into clinical benefit in the clinics. And that's where I'm excited about it, because I want to bring it out and to the patients here. We have a national cancer grid which covers all the cancer centers across India. And if we have this test once completely validated and we have it available for a clinical trial, running it through such a large number of patients would kind of cleanly tell us whether or not this is going to be useful in the clinic. And as I see it, it's an extremely promising test.
Great. Thank you all. This has really been very fascinating. I know there's still a number of questions, but we are coming at the top of the hour. But I did want to say that people from our community wanted to reach out to you. What would be the best way for them to reach? Because as I said, we have a lot of startups in our midst, and maybe somebody wants to take this and run with it. Or maybe there are investors that are looking to take this and take it to the next level. There are a couple more questions, actually. Somebody did mention that, are there any publications that can you post links to publications and information about this collaboration? I don't know, would you be able to point them, or would you be able to share your email addresses so that this dialogue can continue with these individuals that are attending today?
Yeah, no, absolutely, Dipty. I mean, I'll just mention my, I've got a social media account. It's very simple. It's at Anant M, ANANTM. Obviously, I'm reachable on LinkedIn. I think you've got our email addresses. I think all of us are more than happy to provide our contact information, and looking to explore potential collaborations. A lot of publications, a lot of papers, but a lot more exciting work that's hopefully going to be coming out pretty soon. A number of publications that are currently under review, and papers that we'll be submitting out for review. So I expect that there'll be another tranche of publications coming out soon. But we certainly have a lot of work out in the public domain that we'll be happy to share for anyone interested in learning more.
There are a couple more questions, but I don't know, does any one of you want to take?
I could, yeah. Hi, Mohan. Nice to see you again. So Mohan and I go back, we collaborated, I think, over a decade ago when Mohan had just founded Bioimaging, a digital pathology company. So in some ways, Mohan was my first industry partner in the digital pathology space. His question here is about correlating Oncotype DX and pathology imaging. And so that's a great question. In fact, we've done quite a bit of work, several publications now where we've done an association between the image features from the pathology as well as correlating with the Oncotype DX risk categories. We actually had an abstract at ASCO a couple of years ago where we actually showed that the IBRIS test on ECOG 2197, which is one of the completed clinical trial data sets that was used for validation of Oncotype, that we could actually improve the performance of Oncotype by 20%. So essentially using the image-based risk score allowed for the Oncotype DX to essentially identify 20% more truly low-risk patients. So patients who could actually have successfully avoided adjuvant chemotherapy, but by Oncotype alone, they were not identified. By inclusion with IBRIS, or the image-based risk score, we were able to identify 20% more patients. So yeah, again, we've got quite a bit of data, some very compelling data showing potentially also now a competitive advantage over the molecular tests. We've talked a lot about being cheaper, being non-destructive, but at the end of the day, you really want to have the most accurate test possible. And so we've got some very, very compelling data now suggesting that we actually might have a competitive advantage, not just technological advantage over some of these assays.
So one last question, and maybe it'll be good for each one of you to address this. As clinicians, what would you need to see from the AI to feel comfortable in prescribing this use for altering treatment, management for a patient, etc.? So do you feel comfortable? Do you think it's ready to be out in the world?
I guess I would still want to run it through a clinical trial with large number of patients, because we are really ending up, either offering too much of a treatment or denying some treatment, and we definitely don't want to be going wrong there. So we have a very promising tool that's coming up and it's showing us good results, but I would definitely love to have it validated in a larger clinical trial, and that kind of prove, goes to prove beyond any doubt that it is cleanly applicable, and we would be not harming a patient at the end of using such a tool to take a decision to deny a treatment. So that's where I look at it, and I think I'm very happy with the way it's shaping up. Yes, but I would definitely want to run it through a clinical trial.
Yeah, I would just say the guiding principle in biotech discovery and medicine is data is king. Without data, it might be fancy. None of us are going to do it. Nobody's going to pay for it. If you have great data, everybody's going to do it because the data talks. So a good biotech company, a good biomarker company needs to be able to run good trials. I was just on the phone with a different one yesterday, trying to convince them to invest resources in a different disease to run trials, because without which you're not going to move docs and you're not going to move payers. But with good data, it's not even, there's no effort involved. It just happens naturally.
Yeah, so I mean, I could just chime in there very briefly as well. And I think that one of the things that I've learned working with clinicians like Dr. Parmar and Dr. Koyfman is also, particularly the validation, like Dr. Parmar said, the validation specifically on clinical trials, right? You really need to validate on the clinical trials. And that's why this partnership, with Dr. Parmar, Dr. Koyfman is not, you see a lot of these partnerships where, you got a data scientist, computer scientist, trying to get some data from a clinician. I think our audience today would have got the sense that this is a real substantive partnership, a real substantive relationship. You can sense the enthusiasm of the clinicians. So we work very closely together. And with Dr. Koyfman, we one of the first things we did when we started to think about developing this assay for oral cancer and head and neck cancer was, not about the technology. Dr. Koyfman, he is involved with a large number of clinical trials. He's closely involved in the NRG, which is one of the large cooperative groups. And he said to me, Anant, look, these are the clinical trials that have been done. And this is the data that we need to validate the technology. So one of the first things that we started to do is identify what are the completed clinical trial data sets that we want to go after. And systematically over the last year and a half, we've been going and making the applications to these cooperative groups to get access to the data. And that's what I just mentioned. This email this morning suggests that we finally have access to the data. Everything takes time. Nothing is easy. Everything takes time. But the beauty is that the platform's now been laid to get access to those clinical trial data sets. And so similarly in breast cancer, we've had some recent success getting access to an important SWOG, that's a Southwestern Oncology Clinical Group clinical trial. You heard Dr. Parmar talking about the national grid in India. So one of the things that, like Dr. Shlomo Koyfman said, data is king, right? So the more data we're able to put out, the larger the validation studies, multi-institutional validation on clinical trials. That's, I think, when the rubber is going to hit the road. That's when the clinicians are going to say, yeah, this is now compelling enough, this is convincing enough that, I trust it, right? So I think that's the journey we are on right now.
So that's it. We heard it. Data is king. We need more of this, but it's really, really impressive and really interesting to hear that AI technology is being used to transform healthcare. One example over here, we've heard about it for diagnostics, and this is a clear case where therapeutic management can be improved using AI. Of course, more data is needed, more studies are needed, but looks very, very promising. So Thank you all for sharing your time with us today, and we will share this information. This event has been recorded, so we will share it with the rest of our network. And also, I know all of you have shared your email addresses over here on the chat, so in case anybody wants to continue the dialogue or write to you, they can do so. So thanks again. Thanks, Mohini and Vijay, for making this possible. And thank you, Dr. Anant Madabhushi, Dr. Vani Parmar, and Dr. Shlomo Koyfman for taking the time and educating our audience on all the exciting work that you're doing.
Thank you.
Thank you.
Bye-bye.
Thank you.